is John has a sort of unique set of skills and insights that on the one hand, he combines experimental and theoretical work throughout his career for many years. Uh, but then on the one hand, he looks at the sort of the microscopic aspects of learning and memory, like what are the molecular mechanisms of learning and memory, even though I don't think you're going to talk about it much today. And then he's also looking at, in the meantime, at sort of the system level aspects of learning and memory, which is, so this is a pretty broad perspective to take on this very fundamental aspects of brain and behavior. So John is one of the few people around in the field and outside of the field with that really broad but also very strongly experimentally grounded view on the brain. And it's also for those reasons that he can afford to write papers like with titles like How the Brain Works. So John, it's really wonderful that you're here. Thank you very much for sharing your wisdom and insights with us. I'm looking forward to your talk.
Thank you. Well, one of the nice things about getting older is that you can afford to take on some really big ideas. And I think that it's wonderful that neuroscience is the kind of field where big ideas are still possible to investigate. Sometimes you get involved in a field where everything seems to be understood, but in neuroscience we're probably pretty close to the beginning. And so lately I've gotten interested in some very large-scale questions, trying to imagine how an entire group of brain systems would function together to result in an interesting piece of behavior. And in particular, I'm interested in the hippocampus, the basal ganglia, and the motor system. So that's a pretty large set of structures that I'm going to be talking about today. And I'm not going to overwhelm you with a lot of detail. What I'm going to try to do is to sort of make some general arguments about, from an engineering perspective, if you will, how you would get these systems to work together. And just to give you a little bit of a preview, at the level of the hippocampus, I'm going to sort of start with a pretty fundamental issue. What do different parts of the hippocampal system do? And some of you may have heard about grid cells, and then different parts of the hippocampus. And there's been a revolution just in the last year that requires us to sort of re-examine how these different parts of the hippocampus work together, and I want to tell you about that. And then I'm going to turn to an interesting, I think, development that David Reddish, who was here yesterday, discovered. And it's pretty amazing. I mean, if there was one thing that you could say really great about research in the hippocampus, is that we actually can observe memories being replayed. So that's a pretty big deal. I mean, that if you want to study a memory structure, that you can actually watch it do one of its things and try to figure out how it works. And one of the things that has been discovered now that you can watch memories being replayed in a behaviorally interesting context is that all of a sudden it becomes clear we have no idea how to deal with the fact that information is coming out of the hippocampus serially. So first it says, well, this is what happens if I go left, and this is what happens if I go right. But we're not used in neuroscience to think about serial processes. And so all of a sudden we have to think about, well, who's using this information? Well, it turns out probably the basal ganglia is using it to make a decision. But there's really been very little consideration about how the basal ganglia, which everybody thinks works as a kind of parallel computer, how does it deal with serial information? And then when I'm going to very quickly switch to a problem in the basal ganglia. So I was sort of thinking about how the basal ganglia works in terms of a machine. And, well, what is it supposed to do? it's supposed to take reinforcement and convert it into action selection in the future. So if I reinforce when the animal goes right, then as a result of the changes in the synaptic structure of the basal ganglia, next time the animal will go right. Okay. And the reward element in this, the fact that I rewarded it, is important. Anyway, so thinking all this through, how could this actually work? If I was an engineer, how could I build it? Came to the conclusion that there is no way to build this thing without having actually two phases of basal ganglia function. That is to say, first it has to concentrate on action selection, and then it has to deal with accepting the reward signal. And there's no way to make these things work simultaneously, I will argue. And therefore, it has two phases. Okay, you can say, that's not such a big deal. Okay, the structure has two phases. But I will argue that as a result, we have a really radical thing to say about the motor system, which is that the motor system must deal with a structure that has two phases. And as a result, I will argue that the motor system is a non-continuous system. This is really a big deal. It doesn't sound like a big deal to you. But that the motor system doesn't operate with real-time feedback in a continuous way. Rather, it says, okay, for the next 100 milliseconds, I'm going to do this, and now I'm going to wait, and then I give out a command another 100 milliseconds later. That's a discontinuous system. So, I mean, this couldn't be more fundamental. I'm not saying it's true, but I am saying it's fundamental that if the motor system really works in terms of discretized output. So that's an outline of the talk. So let's see how far we can get on this. And so let's start with how the hippocampus works. And I'll explain to you in a minute, I'll show you in a minute what grid cells look like and what place cells look like. These are the building blocks of the system. But what I just want to show you in this slide is that the whole idea of how the system works is, had a previous reigning paradigm, and that paradigm has broken down. And we have to recreate the field. And I'm going to try to recreate the field for you. So remember that the hippocampus is sort of the highest level of the memory system. And right before you get to the hippocampus, you get to a structure called the entorhinal cortex. So the entorhinal cortex feeds information into the hippocampus, and it was in the entorhinal cortex that it was discovered these grid cells. And everybody, including Paul and myself, thought that therefore the grid cells would drive the properties, the most fundamental, simple place cell properties of the hippocampus. Now I'm going to show you that that paradigm has broken down. And therefore, we have to come up with some other idea about what grid cells would be good for. Okay, so let's do that. All right, so finally here you get to see some data. So I'm a rat, I'm walking around, and I have an electrode on a grid cell. And so, brrp, and then it gets quiet, I go over here, brrp, I go over here, brrp, I go over here, brrp, and all of a sudden you notice, hey, every time I go brrp, it's in a little spot, and those 4 spots form an hexagonal array. So that's the idea of a grid cell, and here you can see all the spikes and the fact that they occur in evenly spaced points in the environment. So this is a square environment. This is where the spikes occur. And this is just a color-coding way of showing it. So these are grid cells. And it was very exciting to see cells that had this kind of funny response. They respond at several different points in the environment, and those points are arranged in a hexagonal grid. Okay, so in this recent paper, they found a way of getting rid of the grid cell, or at least the griddiness of the grid cells. So we won't go into the details of how this worked. They injected a drug into part of the brain, and the important point is that now you don't see grid cells. So if grid cells were the thing that was driving place cells, then we should see in this manipulation a loss of the place cells. So now let's look, put the electrode, instead of putting the electrode in the entorhinal cortex, let's put an electrode in the hippocampus where we find the place cells and see whether it's true that when you abolish the griddiness of grid cells, you abolish place fields. Okay, so here's the experiment. And so this is a nice place cell. So you see, instead of having lots and lots of positions that cause the cell to fire, there's just one position. And different cells will have that position in different parts of the environment. So that's a place cell. Now let's do the same manipulation that got rid of grid cells and see what happens to the place fields. So these are different cells, same place field. So this is where the manipulation occurs. And does everybody see the answer? Pretty amazing. Is it clear to everybody? Can you see what's happening? That virtually nothing is happening? So Paul and my great paper, another great paper into the trash bin, right, Paul?
So now we're going to write another one.
Okay, now we're going to write another one. So, I mean, we weren't the only ones to have fallen into this trap, and many of our arguments are still valid, but we definitely have to do a revision of our thinking. Okay? Everybody happy with that? Except Paul? Okay. All right, so now you'd say, well, according to the old theory, right, the grid cells were driving the hippocampus. So if you inactivated the hippocampus, the grid cells should just be happy because they come before the hippocampus, right? Wrong. Okay, so everything is wrong about the old theory. So here we see a nice grid cell, and now they inactivated the hippocampus. Okay, old theory, nothing should happen. Reality, grid cells go away. So it looks like the grid cells don't drive the hippocampus. The hippocampus has something to do with making the grid cells work. So how on earth are we going to fix this? A way of thinking, okay? This is a problem. And I'm going to suggest, what I think is a pretty nice and plausible solution. All right, so...
I could still argue in defense of our paper that, being lazy, that there's still spatial information in those grid cells. It's however reduced.
Okay. Okay, so Paul is right. In other words, well, actually it isn't possible because actually when you look to see what happens to the grid cells, it's not just that the spatial information is reduced, but the spots actually change their position. And that our model would not tolerate. In other words, our model would have tolerated a few spots dropping out, and the residual spots could have been the important one. But our model could not tolerate actual moving of spots. That would definitely destroy things. Okay, I won't give you the right to rebuttal. Okay, so how are we going to make a new model? And I'm going to have to explain these ideas a little bit to you, but let me just summarize it by saying that what do you want to use your hippocampus for? Well, you want to use it systematically to recall stored information about the past. And although David Reddish probably wouldn't agree with this, the past is the future. Why is that? Is because memory wouldn't be of any use to us operationally if things didn't repeat in the world. So, I mean, it's true, we might be nostalgic. Oh, isn't that nice, you know, I met so-and-so. But the real utility of memory is basically not making the same mistake twice. So I went down this alley, and last time there was a wild dog, and so I replay that. Oh, I go down this alley, and I met a wild dog, or I go down this alley, and I found a water fountain, and I felt so much better, and therefore I'm going to go down this alley, or therefore I'm not going to go down this alley. In other words, the underlying assumption of memory replay is that it's a prediction about what would happen if you did the same thing you did before. So I view replay as prediction. It's not absolutely 100% correct prediction, but it's as good as we can do, and it's very valuable. All right, so how do we actually perform replay? And I warn you that there are going to be several different ways, and I'm going to kind of conflate these in the interest of simplicity, and we can come back to that. But at least one way is what I call mind travel. Okay, so what is mind travel? Well, supposing I'm here and I force my brain in a kind of map to move over next to Paul. And, you know, Paul is upset because, you know, or whatever, I insulted his model, not my model. And so I'm afraid he's going to hit me. So I'm not going to go over in that direction. In other words, that position in front of Paul, I associate, with something bad, so I'm not going to do it. Well, that's incredibly important. I mean, the same thing for a rat. It wants to project itself into the future and ask, if I was in that place ahead, would I find a cat? This is very, very important, right? Or would I find something good? So that you want to have this projection ahead to test whether any of those positions ahead of you are associated with something good or bad. Does that make sense? Yeah? Okay. All right. So it turns out that that the hippocampus is constantly doing this every bit of the time, sort of sending out mind travel ahead, and I'm going to show you how it does it.
But John, just to be clear about this, so in some sense now you're splitting up the system because traditionally the idea was, okay, there's a path integration system that keeps track of what you have been doing in the recent past, and you use that information presented via your grid cells to drive your representation.
Okay, so this will become clear. Yeah. So I'm going to have to explain, among other things, what path integration really means, and that's a very important point. So let me get to that. Okay, but first, before I explain how you use path integration to explore the environment or even know where you are, I want to show you the evidence that the hippocampus is producing exploration of the region in front of you. And the way this traditionally has been studied has to do with a phenomenon called phase precession, which is the world's best example so far of something called phase coding. And the phase refers to the fact that when you stick an electrode, a gross electrode, in the hippocampus, while the animal is running, you find that the overall system is clocked by an ongoing oscillation in the theta frequency range, which is somewhere in the range of 7 to 10 hertz. And so now we're going to see what happens when the rat crosses through the place field of a cell. So this is time, but the animal is running, so it's also position. So notice here, the animal is running, and there are no spikes. Here are the spikes. Everybody see that? And now, all of a sudden, it gets to this time, which is also a position, and from then on, it goes, and then finally, it stops firing. Why? Because this position is where the place field is. So I've shown you that, the cell only responds when it's in a certain, part of the environment, the place field. But now you can see it in time. So you can see to cross the place field, they were about 10 theta cycles. Now what John O'Keefe discovered, which is called the phase precession, is this. Look at this spike. It's kind of late in the cycle. These spikes are a little earlier. These spikes are a little earlier. These spikes are much earlier. So there's this systematic tendency of spikes to fire earlier and earlier in theta phase as the animal progresses through the place field. And here's a plot of that. So again, this is now position. So here's the place field. No spiking out here, no spiking out here. And as the animal progresses through the place field, systematic change in phase over about 180 degrees. And then when it gets near the end, something else happens, and we'll talk about what that is. Why there's a sort of systematic lack of interest in phase at the end of the place field. Okay, so why does this relate to the idea that the rat is constantly projecting itself ahead of where it is? So the next two slides will explain it. Now, recently people have actually been able to see this because they can record from hundreds of cells at the same time. But it's only recently. And then this gives, so let's say you could record from a lot of cells whose place fields were very nearby. So to do that, you have to sample zillions of cells because most of them won't have place fields nearby. And so what you'd find is that during just one theta cycle, one cell, which represents the current position, this is a much shorter time. This is just one theta cycle. One cell would fire bum, 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 and then when you get to the second half of the theta cycle, there'd start to be a successive firing of cells with different phase. And so this is what you should think of as the exploration of the environment. In other words, I'm pushing the cell that represents some place downstream to fire, even though I'm at position 1. And then but the change is gradual. So that is the process of pushing ahead. And now you might say, well, how does this relate to the phase precession? Is this really the same phenomenon as the phase precession? And the next slide, I'm hoping, makes this clear. So you really have to concentrate because this is something that always confuses people. Okay, so what we're going to do is to look at the picture I just showed you, but we're going to do it over 2 theta cycles. All right, so we just did it for this theta cycle. Cell 1 represents where the rat is at, and then in the second half of the theta cycle, you get this chaining process. Next position, next position, next position, next position. So now, cell that represents somebody way down at the other end of the room is firing. Why is that important? Because that cell way down the end of the room might be associated with cat, and so I've just discovered that there's a cat down there based on my previous experience, and so I think I will stop. I do not want to go and find that cat. All right, but how do you get the phase precession? Okay, so let's go to the next theta cycle. And the crucial thing is that the phase precession requires that the animal actually have moved. You don't get the phase precession unless the animal moved. And let's see what I mean by that. Well, now we're going to do the same thing. During the first part of the theta cycle, the cell that fires is where I'm at. Well, because the animal moved, you're no longer at position 1, you're now at position 2. Okay, so everything's the same now. Cell 2 fires during the first half of the theta cycle. Then you get this chaining process. So the next cell to fire is cell 3, then 4, then 5. Again, you're looking ahead, and now you're actually looking ahead even further to position 7, to position 8. I should have drawn it here. The crucial point is to look at this. So let's look at cell 3, and what you can see by this slanted arrow is that it now fires with earlier phase. And that's because you don't have to project as far in the sequence because you've already moved your body. So since you're at 2, projecting to get to 3 is not as long a process as if you were at cell 1, position 1. So that is why this combination of the sequence readout ahead of you plus movement of the rat yields the phenomenon of phase precession.
I have a question about this. Is it possible that the place cells, as well as encoding position, location, may also have some mechanism for encoding velocity or acceleration as well?
Yeah, so the question was, do they code velocity and acceleration? And
or some mechanism around them might code velocity acceleration as well, and this might be a side effect of that, perhaps.
Yeah, I don't think it's a side effect, but I think you can get out information about velocity. There's a little bit of information.
Because that might produce a phase precession in some circumstances if you were encoding velocity and position with the same.
Well, okay, so I'll show you some more data, but I don't think so.
All right.
Anyway, why did I go through all this trouble? Oh, let me just finish this slide and then we'll... why did I go through this trouble? So the new evidence, consistent with the hypothesis that mind travel, this travel ahead, is produced by grid cells. So this is the big point. If you write down one thing, Lisman thinks that the function of grid cells is mind travel, to move you through space ahead of where you are. It's not to determine where you are, it's to move you artificially through space so you can test whether a cat is ahead of you. Okay? That's what I think the grid cell is doing. Okay, so what's the evidence that I'm going to base this on? Well, so there's this grand finding of O'Keefe of phase precession, which I just showed you, could be interpreted as this look-ahead process. If you get rid of grid cells, do you get rid of this look-ahead process? So this, not definitive experiment, but suggestive experiment, lesion of the medial entorhinal cortex still preserves a lot of place cell function, but totally gets rid of phase precession. You are no longer looking ahead. It's not a perfect experiment. We'd like a more specific experiment that just got rid of grid cells, but we don't have that. So this is just an abstract from SFN last year. On the other hand, if you don't get rid of grid cells per se, but you get rid of their grittiness by the manipulations I talked about earlier, then you do preserve the phase precession. So you don't have to, there's something about the grid cells which are moving you through space, even if you don't have the grittiness present. All right, so the hypothesis that I'm going to push is that there are ways of knowing where you are, and those ways of knowing are a spatial integrator. This is what Paul is talking about. I'm going to have to, but that is not the grid cell, because you still know where you are without the grid cell. Why? Because we saw that the place fields were normal. The place fields show you that you know where you are in the environment, and you didn't need the grid cells for that. So there's an integrator of space that influences place cells that doesn't require grid cells. On the other hand, what the grid cells may do is an integration operation which allows you to project yourself through space. So what do you have to integrate to project yourself smoothly through space? Velocity. If you integrate velocity, right, you get a smoothly changing position. So I'm going to claim that grid cells are in fact integrators. You put a velocity signal in, and that moves me through space, so I can find out, eventually I get over here and I can find out, what mood Paul is in.
No,
Okay. So this mind travel requires an integrator to move me through space, not real space, but mind travel. And I'm going to argue that that's the function of the grid cells, to artificially move me through space so I can test hypotheses about the positions ahead of myself. Okay, so now let me turn to the questions.
So my question was about the slide where you show that actually the phase precession is due to this replay, but there somehow you assume that the cell are somehow synchronized with the rat because you have a change in place cell every theta cycle, but you will have more than one theta cycle on one place cell before you go to the next. Does it still, so this is the point I don't understand, how does it still holds if it takes the rat longer than just one theta cycle to cross the place field?
No, I showed you a picture, actually. Let's go back and look at that. To cross the place field this place field, it took almost 9 theta cycles. So at the beginning, you're making a projection, ahead of you, and that's what this is. I'm sorry. And then on the next theta cycle, you're starting. It'll fire a little bit earlier because the projection about the position that this cell represents, which is actually down here, is now, closer.
But looking at the graph, this looks now very symmetric. So there's also, you would also have something like a post-play, not just a pre-play. Would you see something like that? Because you also have it also fades out. It not only fades in.
Nope, because I believe that this cell is representing this position. That actually, the position that is, when you think of this as a sequence operation, and this is not the way a rate coding person would think about it, but a phase coding, is that the actual position that this cell is talking about is here. So it fires here with very late phase, meaning that I predict that I'll be at x, I'll predict that I'll be at x plus 1, I predict I'll be at x plus 2, and then I predict I'll be at x plus n, and x plus n is this position. So I'm making a long-range prediction at this point about coming to this position, which is Paul, let's say. And the reason that the firing occurs with earlier phase here is that I've already gone through this distance, so I'm much closer to Paul. So, I only need, let's say, 3 cycles of prediction to get to Paul, whereas if I was here, I needed 6 cycles of prediction to get to Paul. This is very hard to understand, but that's the best I can do. Yeah.
Clear to me if you are assuming that the moving from position A to position B in the field is only possible by a single trajectory. In the open field, there are many solutions to move yourself from where you are to pole position. So it's really...
Yeah, so the simplest way of making the data look simple is to do this on a linear track, which is what most...
Is there a rat in...
But, you can do the same thing in the open field, provided you sort your records to be linear motion. If the animal is going like this, Chris, this way it's going to be predicting this, then it's going to be predicting this, the data becomes very intractable.
Okay. So there is no evidence of a jumping code. I mean that I'm just projecting myself in the future position without the need to track the linear trajectory.
No, it is a linear trajectory. It's a smooth linear trajectory. It's saying within each theta cycle, it's a projection to here, then here, then here, then here, then here. That's shown in this slide here. So this is just one theta cycle. So in this second half of the theta cycle, it's a smooth projection. You know, maybe every 20 milliseconds you say, well, I'm here. 20 milliseconds later you say, I'd be here. So this is a very fast projection forward. Shoo! Pushing ahead to find out whether there's a cat there.
But John, you make two testable predictions, right? Because this would mean that if I would look at the sweeps that you might see in David Radish's data, that I would always see later in the response, when I'm cruising through an environment, that cells are firing of positions I haven't visited yet. So it should be clearly in that data. Right? And let me see.
No, of course it goes all, I mean, this is involving some sort of memory, so you've gone left or right before, but sure, you project, he gets all the way to the goal site.
No, wait, but if you would unpack this, so now if you are in the first phase of the whole story, and you take the place fields of cells 2 to 7, then you're predicting that these should be somewhere projected in a region of space that the rat might visit in the future. Right. So it's a testable prediction.
No, it happens. It's not
so
testable.
No,
that's what it shows.
This is the data we have to see then. How
that is
accurate
what the
that
phase
right?
precession shows. That cells that have their place field centers far out will, further out, will fire late.
Exactly. So this is, if you want, a redescription of the sweeps Dave was showing us yesterday.
Okay.
Right? But,
So
there's a second point that so you're switching around the causal structure that that many people believed in, right? That it's really like you go from cortex and entorhinal cortex grid cells feed forward through the hippocampus to get to your representation of place. Now you're switching that around. Right? You're saying, no, you go from your place cells back into your grid cells to have this ability for mental time travel. But now in some sense you have created a new problem. Because where does then this information on location, on space originate?
Fine. Okay, let's go and do that now. Okay, so I'm going to answer Paul's question now. Now, you remember it was very funny that during the first half of the theta cycle there wasn't any mind travel. You just sort of everything was stationary. And it's only during the second half of the theta cycle that cell A will stimulate cell B, will stimulate cell D, and you do this mind travel business. What is that first part of the theta cycle about? So I'm going to argue that the first part of the theta cycle is taking all available information to determine where you are. Now, what are the available sources of information? So this is what Paul is talking about when he says you need some integrator. And what we mean by that is that, when you get up at night and you go to the bathroom, you know where you are. You're at your bed. But the fact is, and you can try this, you can get to the bathroom pretty well with your eyes closed. Or even if it's dark enough, you can leave your eyes open. So how do you do this? Well, the idea is that you integrate information literally to calculate position. So what kind of information do you have? Well, you have your inner ear which measures acceleration. And since you've all taken Physics 1, you know that if you integrate acceleration, you get velocity, and if you integrate velocity, you get position. So when Paul says, the path integrator, what he means is that if you know where you start, just integrating that inner ear information will tell you roughly where you are. Now, of course, there are other sources of information, like, everything is always complicated. In real life, we also get visual information that helps us know where we are. And so, counting, knowing where you are by the basis of double integration of acceleration is kind of dangerous. You might think, you've gotten to the marketplace, but in fact, your integrator is not that accurate, so you want to look to see if you are at the marketplace. So in the end, you're going to want to combine all available information. If the lights are out, you can't get information visually, and so you would rely on your path integrator to tell you roughly where you are. And in answer to Paul, I think that there are two integrators. One integrator is this integration of acceleration. So it gives you a rough approximation of where you are, and exactly where that integrator is, I don't know exactly. It's probably in the entorhinal cortex, or maybe before the entorhinal cortex. And that is that final stage of second stage of integration is integrating real velocity as determined by the integration of acceleration. So what are grid cells doing? They're integrating imaginary velocity because it's an imaginary movement through space. But the underlying principle is the same. In one case, it's a real velocity signal that is going to move to the position about where you think you are. And then you have another integrator which says, given where you think you are, now let me make you an imagined movement through space so we can test whether there's a cat out there. And that integrator which does the imagined movement through space is what I'm going to suggest is the grid cell. Is that clear, Paul, as a hypothesis?
But it sounds very unspecific, right? Because the grid cells are not necessarily telling you anything about your goals. It might be a machine that allows you to make these projections,
Yeah.
but you have to have a bias on that, because otherwise you're going to explore all possible trajectories, which might not help you. So how would you get the specificity?
Okay, well, I mean, in general, what the data show is that you explore the directions that are directly ahead of where you're going. In other words, if I'm going this way, I'm going to test hypotheses that the cat is there. I'm not going to test hypotheses that the cat is there if I'm going this way.
No,
So,
but take the data David showed us yesterday, where I have this looking ahead in a maze that's very goal-oriented, right? It's looking ahead along a very specific memorized goal location.
right.
So that's not necessarily just a linear projection of the current heading direction.
Okay, so now Paul is getting much too smart, because I told you that
Aha!
that there were slightly different kinds of looking ahead. And I will have to address that complexity if Paul forces me to. So I admit that I've tried to simplify things. So there will be different rules for how to look ahead in slightly different situations. But they may all involve the grid cells to some extent. So now I want to, go to one of the sort of triumphs of computational neuroscience, which tells you how to build an integrator. So if I had a velocity signal, what kind of neural network could I put that velocity signal in order to get an idea about where I was, to get position? And so this is called a bump attractor, or sometimes called a ring attractor. And here's the idea. So first of all, the idea is that there's a particular way in which activity can sustain itself in a bump. And so the bump just says that if you have a network that is this long, there would be this bump. And what this bump signifies is the rate of the firing of the pyramidal cells. So it says that if I could look down with, let's say, a voltage-sensitive dye on this network, I would see very active cells here. Okay? So, and that would be relatively stationary, that bump of activity. And it would be stationary in time as a bump because the cells, the network is arranged for the cells to excite each other. So that's why the bump doesn't go away. It just sits there. A bump of activity can sit there. Now the second point is that there can be special connections in the network that move the bump. And supposing those connections move the bump from, seems like I've lost, move the bump from here to here. And let's say that the movement of the bump is proportional to velocity. Then, if that is true, the position of the bump in the network represents the position of the animal in the environment. In other words, if I can move this bump in proportional to velocity, then the position of the bump becomes a representation of position. So this is a kind of cute trick, right, about how to make networks do interesting things. So it is thought, and we can argue about some of the details, that since you have acceleration information from your ear, you could put acceleration into a network like this, and now the bump would represent velocity. Then you take the output of that and put it into another one of these integrator networks, and now the bump moves in proportion to velocity, and so it represents position. So that is the reigning idea about how you get to the bathroom. This is not the question of how you explore, whether there's, a cockroach in the bathroom. This is just how you get to the bathroom. Okay? So you move, you determine your acceleration, and you can't see, but your integrator tells you, this is where the door is to the bathroom, and indeed, this is where the door is. So you've integrated acceleration to get velocity, you've integrated velocity to get position, and now you know where the bathroom door is. Now, the other kind of travel is mind travel, which says, now that I'm at the door, I'm going to use an artificial velocity vector into the grid cell to move me through space, and I get, to the toilet seat in my mind. Then that evokes the cockroach, so I go back to bed because I am not going into the bathroom with a cockroach. Okay? So these both kinds of travel, real travel and mind travel, have their own utility. Now, if we're going to do mind travel like this, we're going to put a velocity vector into the integrator, which I think is the grid cell. We're going to move through space. That's great. We're going to find out what's associated with the toilet. But then this happens all during a theta cycle. Then at the end, we have to move back to current position because we're going to do the same thing on the next theta cycle, but we want to start our starting point to be current position.
But you do need a reference, right? To make it work.
Yeah, of course. So here we go. This is just the summary diagram of how I think the thing works. So in the first part of the theta cycle, you want to find out where you are. So let's look at the sources of information. So this here, the blue arrow, is information from your path integrator. This is the one that goes back to the vestibular system. It says, based on my integration of acceleration and my integration of velocity, I think I'm at X. Now, notice that I'm calling these cells the non-grid cells of the medial entorhinal cortex. And we can come back to why I did this, but it's not really so important. So in step 2, it goes over to the hippocampus, and incredibly important thing happens. So a second input into the hippocampus is from the lateral entorhinal cortex, which carries information about visual space. So I think I'm at the door. Now let's turn on the lights. I think I'm at the door of the bathroom. but, since I closed my eyes, but it is helpful to open your eyes and see whether you're really there. Your path integrator might not be exactly right. And so we want to have a system that combines as much information as we have, and that happens in the CA3 region of the hippocampus, where these two kinds of information, if available, will be brought together.
But John, question now. The path integration drives your grid cells, which go to
No,
CA3.
these are non-grid cells.
The non-grid, sorry, they drive
Yeah.
population cells in the medial entorhinal cortex, going to CA3.
Right.
And now
Now we have an updated position based on
right.
visual.
And I'm doing mind travel, right? I'm doing
No, we don't. We're in the first half of the theta cycle. No mind travel yet.
Oh, we're doing the real thing now.
Yeah.
Ah, okay.
Okay. So this is the first half. We're just trying to find out where we are. So we found out where we are by taking all information into consideration. And we send it over to the grid cells, and the grid cell says, okay, I am an integrator too. And you tell me where I am, and I will adopt that position, but there's no velocity input. So I'm just going to sit there. Now you've told me where I am. The grid cell says, thank you very much, hippocampus. You've told me where I am. You've combined visual and path integration information, so I really have some assurance that I'm at X. Okay? And the information is going to go around and around. It's just all internally consistent during this first half of the theta cycle. So notice that, what is this? This is the mind travel velocity. We're not mind traveling in the first half of the theta cycle. We're just trying to find out where we are. So this is off. Now we're coming to the second half of the theta cycle. We're going to turn off the inputs to the hippocampus from the external world, both the visual and the path integration information, because the medial entorhinal cortex already knows in the grid cells exactly where we are. And now we turn on an artificial velocity signal into the integrator. Well, if you put an artificial velocity signal into the integrator, you'll start to mind travel. This is not real travel. This is artificial travel. So now I'm moving over to Paul. Okay? And that's very valuable to do to find out how Paul is feeling about me. Or move over to the toilet and find out, was there a cockroach associated with the toilet?
Inhibit certain input from Mac, wouldn't it be more functional to inhibit motor output to do the mind travel?
Okay, we are not anywhere near motor system yet, so this is just memory. We're not, let me talk later about how this is actually utilized. In fact, I don't have any idea how this is utilized. In other words, if you actually projected that a cat was going to be associated with that place, what would cause the rat to actually not go there? So I'll give you some ideas about this in the second half of the talk, but that's a whole other problem. In other words, how do you couple memory to action? And that's I will touch on that. But I mean, that's great. That's exactly what we have to be thinking about. It's just that we don't have a whole lot of experiments yet. So just a side note, interesting that mind travel is very different in the different parts of the long axis of the hippocampus. And I won't belabor this point, but it's just important to remember that the hippocampus is a large structure. In the rat, we call it the dorsal ventral axis. And it turns out that the mind travel is hugely different in the different ends of the hippocampus. So where most of the recordings are done is in the dorsal, and you just mind travel a little bit ahead. But in the ventral hippocampus, the place fields are enormous. The whole scale of everything is enormous. And so I could mind travel over to the other side of the room. So it's just important not to think of the hippocampus as a homogeneous structure. it has different parts along its long axis that operate at different scales.
And also a different modality biases.
Absolutely. Okay, so now we do come to David Reddish's famous experiment. This is the Johnson-Reddish experiment that I like so much. Remember I told you that finally that we could observe mind travel, memory recall, and that it would immediately tell us something incredibly puzzling about how the brain worked, that there was a serial aspect to memory recall, and that we were going to have to learn how to, when coupling that to action, we were going to have to come up with some hypothesis about how you dealt with the seriality of information. So what David found was that fairly early in training, when the animal comes to this choice point on the maze, if it looked left, oftentimes the decoded activity, which he would decode from multiple place cells, during a theta cycle would sweep further and further along until finally it got to the goal site. So then he would wait and continue to observe these place cells, and maybe 1 second later he would see a similar sweep, but in this case it would go down the right arm. And so one of those arms is associated with reward, or maybe even one of those arms is associated with water, and one of them is associated with food. And so what you want to build is a machine, and this gets to your question, how do you take memory information, build a machine that will use this serially presented sweeps of information, all presented during a theta cycle. Boom, down one arm. 1 second later, boom, down the other arm. Okay, what do I do with this information? So I tried to come up with a plausible model. And so here is a kind of model that sort of was built on some ideas that Paul started in his robot, that actually a memory trace should be thought of as couplets arranged in a sequence. Why couplets? Because there's always a state of the external world, and there's always what you did. And so if you try to recreate a whole trajectory through the environment, it's very efficient to do it in couplets. When I got to the door, I turned right. When I saw McDonald's, I turned left. When I got to South Street, I turned right. Well, if I gave you those instructions, you would get through that set of couplets to the eventual destination. And Paul's robotic work said, well, do that. Maybe that's how you do it. And there are indications that the brain has this kind of doublet. The hippocampus has this kind of doublet architecture. Roughly speaking, it's the medial which tells you, where you are and what you're doing, and the lateral, which is telling you what the sensory world is about. And so you can think of the hippocampus as dealing with a sequence of doublets, which is an experience. So at the choice point, I turned left and I got to drink. That's one episodic memory. At the choice point S1, I turned right and I got to food. So you have these memory traces, and now you've seen that we're able to mind travel through space that would play out these memory traces. But what happens next? And so I was just wondering, well, how on earth do you deal with seriality? And we'll come to the question of how the basal ganglia works in a minute, which does action selection, but the reigning models of action selection are based on parallel comparisons between alternatives. So we have a big problem. We have the information arriving from the hippocampus that's relevant serially, and we have an action selection device that works in parallel. So how are we going to make these two parts of the brain work together? So my proposal is that every time you read out one of these memories, you download the information into synaptic weights that have a long-lasting, that last at least minutes. So if you download one item and put it in weights, and you download the other item and put it into weights, now you have both items, all the information stored in weights, and now you can use a parallel.
That's working memory.
Nope.
No? How then?
Well, it depends on how we were arguing before. It could be called working memory unless you use the definition of active memory as active maintenance, which some people do.
Right.
But here it's not active, here it's stored in weights. But it does decay, and so in that sense you could call it working memory. So it's a temporary download. So what have we done? Well, when we download the left turn, we make a connection between all the different kinds of information. So S1 was the choice point sensory stimulus, which we're going to connect to drink, which is the outcome, and we're going to connect the outcome to left, which is what led us. Then we're going to download, 1 second later, what happens if I turn right. So again, we have S1, but now it's connected to food, which is what was the outcome at the end of that pathway. And we're going to connect food to what we did to get that outcome, which was turn right. So you can follow the strong weights by the thickness of the arrows. So you can see that different connections are enhanced by these two downloadings. Now the really cute thing is that psychologists have determined that at this stage of learning, before things have really become habit, in fact, internal state does make a difference, as you'd expect, as you would hope. So if you're hungry, you're going to do one thing. If you're thirsty, you're going to do another thing. So what we want is a memory readout mechanism that converts serial to parallel. Well, I've showed you how that can occur, because now both weights coexist. But we want to do it in such a way that we can solve the problem of value, which means that the ultimate decision should depend upon my internal state, whether I'm hungry or thirsty. So far I've just read out the history of these two pathways. So let me just show you that it works. So all I'm going to do is say, if I'm hungry, it's going to potentiate this synapse onto food. So remember that the downloading, as you can see, strengthened both the S1 to drink and the S1 to food. But now I'm saying, since I'm hungry, so there's going to be some neuromodulatory effect which turns up this synapse. Okay, what's going to happen? Well, now this pathway is going to be stronger than this pathway, and therefore right turn will be the choice I take. So this is a parallel comparison. In other words, the system down here is looking to make the choice depending upon whether left or right is differentially activated at the same time. That's why it's a parallel computation. And so the combination of these arrows, which are the strengthening resulting from the downloading operation that occurred serially, in combination with this hypothesis about neuromodulatory control, will, in fact, make the animal take a right turn if the animal is hungry and a left turn if you're thirsty. So I believe this incredibly stupid, simple model solves two fundamental problems in terms of the relationship between memory and action selection. Number one, it shows you how you can use standard parallel basal ganglia processes to operate on hippocampal information which was downloaded serially. So parallel processes make the decision based on information that was serially downloaded, and the trick was that each serial download made a long-lasting synaptic weight change, so at the end of both downloading, you had weights that you could compare. The second problem that is solved by this very simple model is how to make the action selection based on current state, whether I'm hungry or thirsty. So I don't know. So I mean, it's just a working hypothesis, but it's interesting, and I actually haven't found anybody who's generated a simple model like this about how to couple the Johnson-Reddish observation of serial downloading into a plausible way for using the parallel processes of the basal ganglia to make the action selection. So, question?
Yes, so when you do this serial downloading, you start with sensory motor couplets that represent each segment of the path that eventually ends in a goal that is either food or water. But at the moment in which the decision has to be made, the sensory motor couplet does not represent what is going to be at the end of the path, meaning that in this moment when you do a comparison, there is no sensory information that food is there or water is there. It's gonna occur later in that chain, but if you do this parallel comparison, in that moment that information is not going to be compared. So
Okay.
something like the goal information is missing. So
Okay,
maybe
so yeah, you just,
you should do the serial thing backwards.
sorry, but you missed the full import of this picture. So in this picture, just during one theta cycle, it sweeps through all the positions to the goal. So there is, you've got the entire picture of that path and what's at the end of it, all downloaded in 120 milliseconds. And then about 1 second later, I was asking David yesterday, how long does this take? About 1 second later, on average, you'll do the other path. So after several seconds, the weights have the entire information about both paths. And so you can operate on that.
Downloading of weights, the realization would be synaptic facilitation. So it's
Yeah, I don't like the word facilitation, but short-term potentiation, like a very short-lasting point version of LTP. Okay, so I'm almost at the end.
John, sorry.
Um,
Yeah, John, just I was commenting about this idea of with Radish, okay, that you could make this forward in the future and used as an integration for different motors. And he commented with me that in this case of when you have the selection, you cannot predict from the frequency of sweeps whether it was going to the left or to the right. And actually he found some, he said that there was somehow more like you're trying more to get information from the part that you actually don't go at some point. So how would you see this would fit, for example, in another model that you're actually accumulating information? For instance, like a race model wouldn't fit well here, like if you had a race model in the basal ganglia.
Yeah, so I think that it has to do with the question of how many sweeps in one direction, how many sweeps in the other direction, and is that important? And I don't know the answer, but I mean, you could certainly sweep in the direction that you're not going to go and find out information that was useful, like there isn't water there. So I don't necessarily see it as a unidirectional kind of thing.
So John, I don't understand why you want to copy the whole of your trajectory up to the striatum, because really all you need is the value of the action of turning left and the value of the action turning right, and you send those to the striatum and it picks the biggest. And you can get that because you have a forward model here, you know what the rewards are, and you can do dynamic programming with this forward model to give you the values of the left and right actions.
No, I agree. So I don't know exactly, how you would design the system to deal with the fact that there's a whole stream of information. As you say, you don't need all these intermediate positions to solve the problem. You really want to know what's at the end. And of course, what's at the end is associated with reward, so that may give it a little bit of a highlighting.
Well, you would argue this gives you, of course, intermediate checkpoints, right, to see whether you're actually doing what you thought you were doing.
Okay, so let me just finish up and then we can have, oh, okay.
I was wondering, young people should sit in the front, not that, then the old people don't have to walk so much.
It was just to make you tired, actually. And don't you have a problem with timings in the conversion from serial to parallel? Because like sweeps take some times, actually.
Yeah, they
the order of seconds, right?
No, 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 no. A sweep occurs in a theta cycle, occurs in about 100 milliseconds.
Okay. And then the parallelization happens like at the order of, what's the time scale of that?
On the order of seconds.
Okay. And
so in other words, you have to wait until you get a sweep down the other side.
And is this consistent with the decision-making literature? In terms of timings, I'm talking.
Yeah, I mean, what David says is that actually, you know, the animal stops for several seconds at the choice point, and so in a sense, this is what's happening.
Thank you.
Okay, so last couple of complicated ideas, but I promised you that, so we have here the sort of standard now model of how the basal ganglia actually works. And so we've been sort of concentrating on this part here, but we haven't been talking about the direct and indirect pathways through the basal ganglia. So here's the direct pathway, and it's considering left and right actions, which kind of funnel all the way through to the actual motor system. Now, the point of this slide is to say, yes, but there's also this indirect pathway, and it's also, that indirect pathway is called the no-go pathway. And the reason is that by the time you sort of look through the various sequence of inhibitory actions, that activation of this pathway will tend to inhibit action, whereas what we talked about before is the direct pathway where activation of the cells through this complicated synaptic arc winds up activating. Well, why do you want to have a direct and indirect pathway? And the current thinking is that the indirect pathway is strengthened by aversive stimuli. So if something good happens, you strengthen synapses on the direct pathway, which means you're more likely, you know, if you turn right, you get reward, you're more likely to take a right turn. But what happens if you turn left and get a shock? Then you want a system that will make it less likely that you make the left turn in the future. And the notion of these models is that the strengthening of synapses onto the indirect pathway winds up inhibiting action. So if you take a left turn and you get a shock, the synapses onto the indirect pathway will be strengthened, but the indirect pathways, because of the extra synapses involved, winds up inhibiting action. So that's why it's called the no-go pathway. Now, what I'm going to show you is that there's a tremendous problem that sort of has been hidden and which is very interesting. And the problem is illustrated here. So if we consider first this case, so here's the direct pathway and here's the indirect pathway, and one of the actions, right, got a big reward, that's signified by the star, and the other pathway got less reward. Which are you going to choose? Well, the whole idea is that this pathway is stronger, so action selection selects R. Cool. And how do we know, how does it work? Well, these cells fired the most, and so dopamine will strengthen them the most. Dopamine carries the reward signal. And so now the synapses onto this are strengthened, so in the future, this pathway will get even stronger. Cool. Everything is simple. The problem is that, you know, in our experience of life, we've tried different things, we've gotten some funny mixture of reward and punishments, and still we make a choice. And so there's some sort of mammoth voting process that's going to say, well, if I turn right, you know, I might have gotten 3 times I got incredible reward, but 1 time I got punished. If I go left, you know, this and this happened, and somehow we integrate all of this information about reward and punishment for the different actions, and we make a choice. And let's look at the tremendous problem here. So here we see the various votings. So the cortex selected R. Okay? Now, which synapses, and R will now get reinforced. So dopamine comes, which cells should it act on? Should it act in proportion to cell activity? So do you see the problem? That if this is if star represents cell activity, we can see why why right was chosen. Why was right chosen? That's right, because the inhibition here canceled this. Does everybody see that?
This kind of answer the question, because at the end, if you inhibiting at some point more important than actually potentiation. I mean that if you are collecting information both about going and both about non-going, at some point it's more interesting.
No, this is not going. Well, this is just saying, this says don't go left. This says don't go right.
But that's fine. At some point, it's interesting. Like if the Reddish was saying it, he was looking more to the part that he was actually not going.
Oh,
so
I
at
see.
some point, like he already made in his mind that okay, maybe it's a good idea to go to the right, but let's check to the left.
Right.
Then he's collecting information about not going to the left.
Could be. But let's just focus on this. I want to finish on this simple point, that here we chose right even though left was incredibly active. And if we now had a rule which said dopamine interacts with the cells that were active, we get a horrible problem. Because it would look like we would reinforce this cell. So that's a horrible problem. What we want to do is reinforce the cells that led to the correct action, but the cells that led to the correct action are not necessarily the most excited cells. So biophysically, how does the dopamine signal know on what to work? And I claim it's impossible unless you do something different. And what I claim is that what happens is that the activity levels here are in the first phase of the overall process where you're selecting action. Now, in the second phase, your brain knows what you selected. So if you selected right, you wipe out these stars and you send what's called a corollary discharge. That is a copy of the actual choice you made. And since the choice you made is R, then this would light up big and this would not. And now the dopamine signal, on the basis of that signal, can give appropriate credit to what you want. So if you just did this the way it is, if you assigned credit to this, it would lead to counter conditioning. You turned right, you got a reward, but you strengthened left, so that would be horrible. So to avoid this problem, I'm saying that in the second phase of basal ganglia function, these stars get erased, and the actual signal about what you actually did gets imposed on here. So if you turned right, this will be active and this will not, and now the dopamine will strengthen synapses on those cells, and that's what you want, in fact, to do. So,
let's create a new problem now, because the stars reflect also sensory states that might give you evidence to make a certain action to the right or the left. If you then rely on reinforcing those pathways with only the corollary discharge, you only have the action information and you don't have access to the sensor information.
You're right.
anymore.
No, you're absolutely
So
right.
how are you going to fix that?
So it has to be some sort of joint. In other
Right.
words, the actual cell has to have been active by the sensory information,
Yeah.
and then it gets acted on by the corollary discharge. So you're absolutely right.
That's how DEC solves it.
What?
That's how it's solved in DEC.
Okay, good. Okay, let me just finish, and then I'm so near.
That is your zone.
I'm so near the end. Let me just finish, and then we'll deal with it. Okay, so this is just a paper, the closest I could find that would provide support. There are certain types of cells, not all cells, in the basal ganglia. So when the action is taken at time 0, there's a big 100 millisecond burst of activity, but only when the cell represents the chosen action. So these are called chosen action cells, and the fact that it occurs after the action, is evidence that there is some plausibility to my suggestion that the basal ganglia gets information about which action was chosen. So I have just 2 slides left, and then we can go to discussion. So the shocking thing to me was when I finished this part of the project, I said, holy smoke. In other words, it seems like I can only solve give the right credit to the cells that chose left or chose right if I have a 2-phase basal ganglia system. First, a phase where that all possibilities are considered in parallel and an action is chosen, and then the second phase where you impose onto the system just the chosen action to interact with the reward signal. Then everything is beautiful. But isn't it interesting that we've been forced now into a 2-phase system for the basal ganglia? It's not a single-phase system, a 2-phase system. So then I wondered, well, if that's true for the basal ganglia, well, what about the motor system? Maybe the motor system isn't continuous either. Maybe it has some sort of 2 phases or clocking. So I started looking through the literature, and I found that indeed there's many people who have suspected that the motor system is discontinuous. And what does discontinuous mean? Well, the specific hypothesis is that an action, a command to the muscles or to the spinal cord only goes out once every 100 milliseconds. And then you can't put out another command until 100 milliseconds later. So it's a discontinuous system. So I just want to show you a little bit of evidence that I think is pretty impressive. So the first bit of evidence were people who just said, well, if this is true, then maybe if I studied what seemed to be a smooth motion, I would actually see in detail that it was not a smooth motion. And in fact, this is exactly, so when you move, this is moving your finger like this, measuring the velocity very carefully, and this is not 0 here, so don't get confused by that, but when you look very carefully, you find that the velocity is varying at 100 hertz. So this is, in this particular experiment, it took a second to move your finger like this, and during that second, the velocity was not constant, but sinusoidal. So that begins to look like a discontinuous system. And there's some very interesting things about the extensors and the contractors, what are they called? Anyway, being slightly out of phase. But what experiment could I show you that would prove it? And I looked and looked for this experiment, and finally, a few days ago, I found it. The authors didn't even quite realize, I don't think, the importance of their result. So what I would say would be the proof to me that an oscillation organizes action is that action can only be taken at a certain phase of the oscillation. And so in this experiment, an electrode was put in the motor cortex and the animal given a flash of light to make an action, but there was no connection between the flash of light and the EEG. But when you look back on the experiment, you can find that there's a latency fluctuation in the action, and that fluctuation is explainable by the phase of the ongoing EEG. So what this slide shows you is that when they look at the action here, that it occurs on a particular phase of the ongoing oscillation, which is just averaged here over many trials. And you can see that 200 milliseconds before the action, if you're looking at the EEG, you can predict when the action will occur because there's an ongoing oscillation with a certain amount of regularity. If you know where you are in that oscillation 200 milliseconds before, 300 milliseconds before the action, you can predict when the action will be taken. So in my view, this is, the strongest evidence that I can find, and I think pretty damn strong evidence, that there is an oscillatory character to the whole motor system. And this would be incredible. I mean, if we had just agreed on this principle, I'm not saying you should agree on it, but if it were really true, then everything you think about how to design a motor system is going to have to struggle with this fact that there's a cyclical basis for motor control. So remember, I told you in the beginning that we were going to struggle with some pretty big ideas here, and I think this is one of them, that when we finally come up with models of motor control, it's going to fundamentally have to be an oscillatory model.
And you're saying this is a signature of this two-phase model of basal ganglia?
Well, at least it's in the same spirit. Right. That, so now you have the possibility of dividing 100 milliseconds, and you say, well, 50 milliseconds I'll devote to, or some fraction of that cycle I'll devote to action selection, some fraction of that I'll give the feedback of corollary discharge. So these are compatible ideas, is the way I would sell it. So just my summary slide, just to reiterate the points that I've made. First of all, I'm trying to reinforce the idea that we have seen memory sequences being produced, that the hippocampus is all about sequences, that sequences allow you to predict what will happen. It's incredibly valuable to predict what will happen. And this prediction process is at least in part due to the grid cells, I claim, that their function is not to do the integration of position, of real position, which is done by some other integrator. The grid cells are responsible for mind travel. They are moving you artificially through space so you can find out, what's ahead of you. Then we talked about this fact that the observations of serial readout, first down one path and the other path, and that this seriality could be simplified operationally in the machine, which downloaded the information into weights, making it possible to make the comparison between left and right paths based on a parallel computation, which is sort of the standard model for basal ganglia function. So that's important. And that even then I could sort of wave my hands and make a model of action choice, which took into consideration internal state, which is cute. Then when we took a close look at basal ganglia function, we said, oh my God, to make it work right, we're going to have two phases. We're going to have a phase which does the action selection, but then a second phase where you put the corollary discharge in and you say, this is what I did, And now that I know basal ganglia knows what I did, it can offer the proper credit assignment. And then when I finally had talked about a two-phase operation, I said, well, maybe this is a more general principle. And maybe even the motor system has to operate in a discontinuous way. And I showed you some evidence that that was the case. So I'm tired, I'm finished, and now I'd like to answer all your questions.
To question 4 here, there were a few slides back. You were showing the competition between the inhibitory and the excitatory, and you were saying that only the action chosen should strengthen the weights.
Right, subject to Paul's correction.
Yeah. I was going to ask you. Yeah, that's the one. Let's say you took the action and you actually got a bad result. So you wanted, that would also have to be taken into account in terms of strengthening the inhibitory weights.
Right.
So is there an easy way of adapting the weight update that would take that into account appropriately?
Yeah, I mean, it's exactly the same problem. Of course, the bad actions, according to current theory, have to do with not an increase in dopamine, but a decrease in dopamine. But again, you don't want to necessarily have that dopamine signal operate on the most active cells during the selection. So it's exactly a complementary argument that you need to bring in the corollary discharge to identify the cells that act, that dealt with the action.
You'd want to strengthen the inhibitory weights for R, in that case, let's say. If you made the R decision,
right.
Would you weaken the excitatory?
Well, you probably would, because some people think that the drop in dopamine will actually weaken the synapses. So this is now getting into some of the details of what we know about dopamine interaction with the weights, and it's a little bit confusing.
Okay, thanks.
So you would have the replay during the rat going through the linear track that will have given rise to the phase precession, but then you have the replay at the decision point where the radish experiment. So what would you see the functional roles of these two replays? Will they have different roles? Well, one is for the decision making, but what's the role then at the linear track for doing it?
So, you know, I mentioned that after Paul's questioning that I'd simplified this a little bit, and there are probably reasons to think that what's happening at the linear track is different from what's happening at the T-maze junction. And on the linear track, it seems like you're just always projecting straight ahead. Whereas, as Reddish showed, when you're at the choice point on the maze, you can actually turn a corner. Because actually you have to turn the corner to find the reward site. So what's at issue here is whether the grid cells can get you all the way to the choice point. I mean, to the reward site. And I think they can't. So what's very, very simple is to see, well, I have an integrator, the integrator of the grid cell integrator. I put a velocity signal in. I'm going to go straight ahead, right, as I integrate it. I'm not going to go around any corners. And so that would seem to be an adequate explanation for what happens on the linear track, but not an adequate explanation for what happens at the choice point where you can go around a corner according to the Reddish data. Right? You with me so far? Okay. So here's a possible reconciliation. So you push into the CA3, dentate CA3 system a linear probe from the grid cell. Now, we've already established the idea that, you know, when I hit a certain place field, that place field might be associated with cat, and that's very valuable. That's called auto-association, place and item. But you may hit a place in the CA3 system which stores the episodic memory that at that place I took a right turn. and take you over to this cell. So now you can begin to see how the grid cell system, in a sense, could serve as a cue to the episodic memory system, such that when you got to a point where there was a memory that took you off the straight line, the trajectory, in fact, went off the straight line. So that would then be sufficient to explain what Reddish saw. So, in a sense, you could say that the spatial exploration is some combination, actually, of an artificial velocity vector, which is sort of stupid, which gets you going, but then it can hit, in a sense, a resonance, hit an episodic memory, which then takes over and takes you on a path. That's the best I can do at this point.
In your model, there is no subthalamus, so there is no
No what?
no subthalamus,
No subthalamus.
so no hyperdirect
Right.
pathway. What do you believe is the role of this pathway?
I don't have any particular take on that. I mean, what people say is that, you know, if you sort of look at the signs of the synapses, it looks like the subthalamic nucleus will stop action very effectively, and so there may be situations where you want to countermand, and that would be a pathway to use. I don't know. I don't have an opinion.
Okay, so as you explained, theta phase precession may be an explanation for the sweeps in these decision points, right? But if you take the rat to the open field, because there was this study by Pfeiffer and Foster in 2013, where they were observing the sweeps in the ripple events. So how could this fit into the ripples without the theta rhythm?
Good. Very good point. So yet another level of complexity. The ripples are another form of electrophysiological signature that is different from the kinds of oscillations you see during theta. When do you see the ripples? When the animal seems to be offline. In other words, just the opposite of being at the choice point on the maze. Oh, it's sitting there, it's munching on carrots. I don't know, I guess they don't eat carrots. But anyway, munching on whatever it eats, and or just resting. And then all of a sudden you'll see this sharp wave thing, and it seems to replay a trajectory. Now, what we know pretty much is that that is generated within the hippocampus itself. Does not need the entorhinal cortex at all. So this is yet another form of replay. That replay goes around corners. So it's not a linear thing like phase precession. So that would be consistent with a replay of an episodic memory. You know, I went this way, I turned that way, I went that way. So the going thought is that, you know, there are episodic memories stored, and so in the sharp wave you're seeing those in pure form. It's an intrahippocampal generation.
So John, in some sense, so you looked a bit in detail at this interaction between information we store in hippocampus when we navigate a maze, and then how you can also exploit that information for decision making by interacting with cortex and basal ganglia. But now if you talk about, let's say, uploading this information to this cortical basal ganglia system, in some, there's a lot of stuff you need to explain. And I could also argue, but look, why do you even need to upload this information in this form? Because as the animal runs around and information goes into hippocampus to become episodes, the same information is also targeting these regions in cortex and basal ganglia. It's already there. The only thing that you have to, if you want to upload, are relationships. You don't necessarily need to upload, if you want, the semantics of this. Like, okay, I saw these landmarks and I made these actions and so on, right? So don't you think that the notion of uploading that you're describing, unpacking here, like really taking all this content in hippocampus and distribute it in this cortical basal ganglia system is not a bit, let's say, an overkill, because a lot of the stuff is already there.
Okay, so I puzzled over this. I think I have a pretty good answer, and David even agrees.
So you're saying
so
actually
you're
it's
sunk.
accepted.
You're sunk. You can't be too skeptical. So here's the way. So I mean, the operationally to test your hypothesis, you just lesion the hippocampus and ask whether you can train the animal to do the task. And the answer is that there are many things you can do without a hippocampus. All right, so what can't you do without a hippocampus in terms of conditioning? So some of you may know the classical conditioning experiments. You know, you ring a bell or you make some sort of noise, and then the unconditioned stimulus comes out. Let's use the eye blink. And it turns out that if you take out the hippocampus, the following experiment will not work. So I give a tone, boop, I turn the tone off, and half a second later, I spray, I give a puff of air to your eye. So will the animal, without a hippocampus, develop the conditioned response, blinking, on the basis of the tone? Interestingly, that's called trace conditioning. Will not work without the hippocampus. So why is that? Well, it's because you're trying to connect events over time. So you have the end of the tone, and you have to connect that with an event later, which is the unconditioned stimulus. Okay, so I would like to argue that the same thing is true in the Johnson-Reddish experiment. So if we actually had a situation where the animal turned right and immediately got a reward, then maybe you wouldn't need the hippocampus. You could do the association. But in fact, that's not what happens. The animal has to run down and get the reward. So you need a representation over time, which is a sequence, which is what the hippocampus does. So that's
no, but then we agree, because then the key thing you want to transfer to these other areas are relationships. It's not necessarily the
well, relationships over time. In other words,
yeah.
you can make the relationship of the conditioned, the conditioned stimulus to the, well, to the response. I mean, to the contingency, the unconditioned.
Okay, that's a stimulus, yeah.
If there's no delay, but if there's a delay, you need
Sure.
the hippocampus.
No.
Okay.
Perfect. We agree.
Okay.
All right. Do we have
Good.
more questions? Oh.
I'm really unhappy with the idea of a motor system considered as a discontinuous control for it. Your argument is that at the time when you move to the basal ganglia, the observation of the activity in the direct and indirect pathways following the need to stop or to interrupt a decision or to have a less valid as done before the alternative solution is essentially based on the reactive control or in the go-no-go task. But there are evidence in the literature of the countermanding task that you related before, that the two systems are actually working on parallel. And if I know that there is a stop that is only possible in the future, my go process slow down. That is a clear evidence, to my opinion, to other, that the two systems are working on parallel.
I thought I didn't want to make any statements about countermanding, except I was forced to by the question. It's not part of my theory. So the theory is that, and I think this is very well established experimentally, is that when you take just a typical decision and you put electrodes into the basal ganglia, you find that cells in both the direct and indirect pathway are working at the same time. So there's a voting process, simultaneous voting process for and against different actions. It's that fact which I think poses the problem. That once you admit that fact, then you can't assign credit anymore on the basis of activity alone. That's the problem.
Okay, but this is your argument is that this is possible, but it's not the only way how the basal ganglia works.
Okay. Fine. I mean, I'm not really an expert in the basal ganglia.
The motor system, at least, because
Yeah.
the other point that the motor system requires at least 100 milliseconds to perform a different action could be related to the sensory process or to the motor effector. Your point that it's very difficult to change your direction of a finger. It's
No, I didn't.
It's a filter
I didn't
that's
say. I did not say.
on the
Oh,
periphery.
maybe I did say that. But okay, I did say that, right? I said once you made a command, you couldn't make a countermanding. You couldn't make another command, and you're saying that may be wrong. That you can countermand.
No. I'm referring to the latency of a further command to the motor system that is limited by the sensory system. Because if there is another signal presented,
of
then
course.
the cortex requires at least 70 milliseconds to process it.
Yeah, absolutely.
And at the same time, if the signal is sent to the periphery, to the muscles, it requires a while. There's a filter over there
Exactly.
to be transformed in a movement. So
But in this experiment that I showed, this experiment was recording in motor cortex and finding that the actual action was predictable, 300 milliseconds ahead. So there's no sensory involved here.
which one is the point? Is the
If you look up, in the that noisy trace, which I've amplified, so you see an upward oscillation, like 300 milliseconds beforehand. So from that, you could predict that the next upward oscillation, would be, 150 milliseconds later, and that is when the action occurs. So you have, what's important about it is that it's a phase-locked event. That's all I'm saying. So the action cannot occur at arbitrary phase of the ongoing oscillation.
But I think to understand Stefano's objection, maybe we have to clarify a little bit what really the issue, the sticking point is. Because Stefano, are you saying it is not a discretized system at all, it is continuous? Or are you debating, let's say, the discretization time window?
I think it's no conclusive. That it is. I think that is a possibility that is discontinuous, but.
Okay. Well, I certainly wouldn't want to argue that it's proven, but this I thought was the strongest. And on this kind of experiment, there have been some further experiments that I didn't mention, and so for instance, you can ask whether or not you can find coherence between this peripheral oscillation, which is very, very small, and brain oscillations. And so that's not a time locking, but it's just a coherence argument. And this paper uses MEG to actually find that in the inferior olive that there are oscillations that are coherent with the peripheral oscillation. Now, the problem with this is that people will say, oh, this is just all tremor. And of course, tremor is everywhere, and so that doesn't surprise me. That's why I think this is suggestive, but not proof. To me, this experiment is the stronger one. But I still think that, this is just the beginning of work in this area, so we shouldn't be too firm.
And in some sense, Stefano, your own data might confirm John's point, because with the countermanding task, if the interrupt comes after let's call it some threshold value, you cannot recall that movement. That again, it could be related to the sensory process. Absolutely, but still, it would show that the process is partially chunked, let's say. It is discretized. What the cause then is, okay, then we debate what's causing this. I think the only thing John needs is that then you have these discrete windows. And if the sensory state helps you to create them, then he's home free. That's the problem.
I was going to make the tremor point, and I think there are studies that show you can get tremor without having to have an underlying oscillator. You can get tremor just from the mechanics of the system. But I mean, we all know that there are oscillations in the brain, but what I don't understand is what you're trying to claim here about those oscillations, because if there are oscillations, then the system, the activity of the system is often going to be phase locked with those oscillations. So, but that doesn't necessarily have any functional meaning. And you know, we have an oscillation in the sound system. There's a quiet little buzz that we can hear. There's an oscillation in the lights. So you can have effectively a smooth transmission from our point of view of illumination or of meaningful sounds over and above these oscillations which occur in the system. With the hippocampus, you have a nice story, I think, of how maybe the oscillation is being used to do something computational. But across the rest of the brain, I don't see that you can generalize that.
That was why I thought this slide is so important. In other words, you can say, yeah, the oscillations are there, but they don't control action. But this is the counter evidence. So you may say, oh, it's not important that the action can only occur on certain phases of the oscillation, but I don't see how you can say that. I don't think that's legitimate. In other words, if it is phase locked, then there are periods where you can't act. And if there are periods where you can't act, I mean, then that's a fundamental design principle.
Well, the limitation of the system.
Yeah.
That the oscillation perhaps imposed.
Yes. But I don't see how you can say that it's not important then. I mean, if you can't act in one half of the cycle of the oscillation, you know, this is as important as the gas pedal is to the car. I mean, it's key.
Yeah, but I think that the sticking point is that I think the key of John's claim is that you need any form of discretization in some time window to make his system work, because he just wants to be switching. What the cause is of that of the discretization is in some sense not a key issue, right? The key issue is that you have good discretization at all, and I think that's not really what we're debating. No, it's more the genesis, the origin of these oscillatory aspects of motor control that's now being debated. No, just I feel that's more the issue here than the core hypothesis of the discretization of processing.
I guess I'm just saying I'm not surprised that there are these fundamental frequencies.
But you know, when I talk to people in motor control who don't believe in the discontinuous system, they say, oh, you have continuous feedback loops, and I mean, as an engineer, I would say, continuous feedback loops are great, but I would say the brain is not one of them.
But I mean, we have continuous illumination here, but if you have a high-speed camera, it looks like thicker. So it just depends on the granularity on which you're looking at the systems.
Well, so I mean, we can. There are interesting arguments in the vision field about the discretization of vision. This is how you get the wagon wheel illusion, according to some people. I think that the whole brain, I think the truth is going to be that the whole brain, from beginning to end, is organized by oscillation. So that's my bias. I mean, this slide looks at the basal ganglia and the motor control system as kind of more of an integrated look. So you can see each 100 millisecond period. So I've combined in those boxes statements like, okay, the action selection system, has its chance. It hands it over to the motor system. Now it's going to concentrate on evaluation. It's going to take the corollary discharge. It's going to take the dopamine signal, and it's going to do credit assignment. Now the motor system got its command, and it's going to send out it's going to take all the feedback that arrived in the last 100 milliseconds and say, well, this is my best estimate of where the system is at. This is what the basal ganglia is telling me to do. And now I'm going to send out a command to the spinal cord. And I'm not going to do anything after that for another 100 milliseconds. Now, there may be fast things going on in the spinal cord that can make quick adjustments, but I'm going to get feedback during the next 100 milliseconds, and then I'm going to make my next command. Well, I'm not saying that's true, but that is just an incredibly different design principle than continuous feedback. right? I mean, so we're talking apples and oranges here.
Last question, Armin.
When I first saw grid cells, I was always baffled about the hexagonal structure. Now, you gave a new, and there was no real explanation more than it was the most compact representation, or at least none I would know of. But now you give a new role to the grid cells. Would you see any functional role for the hexagonal structure?
Oh boy, that's a great question. I'm working with a physicist, Marco Idiot. He is always asking me the same question. All right. Well, I think we should do it over beer. I have an answer, but you won't like it.
Well, but hexagonal structure is the most compact description of a sphere, right? But anyway, the very, very last question comes from Giovanni in the back there.
Yeah, I was just curious to know if you can find like the same kind of oscillations that you're talking about in Parkinson tremor. Do you see like a specific frequency or time signature in that?
Well, I mean, you see the frequency of the tremor.
Mm-hmm.
I mean, there are people who argue, that Parkinson's is an exaggeration of a normal process.
Okay.
But your question is a good one, and I don't really know the answer.
Because that could be like something to look at, looking at this like discretization.
I mean, it makes sense that if the system is oscillatory, a lot of oscillatory systems can get over oscillatory,
right?
And maybe that's Parkinson's, but I'm not in a position to answer it.
Thank you.
All right, great talk, John. Thank you very much. Okay, we have a break now, and you remember, right? So we continue now with these sort of road mapping activities for the Convergent Science Network. So after the break, I will start with, let's say, sketching a little bit what we try to achieve in the future of the convergent science of brain and technology. After which we're going to look at more specific statements and predictions of different people running different projects in that domain. The idea was that students or participants in the workshop should prioritize their projects. But of course, if you want to get a bit of a flavor of where we believe, at least with the participants in this road mapping session, where the field is going, you can join this first part of the event. You're absolutely invited to do that, and also to then share your own opinions on these things. So that's what's going on now after the break. Second thing is, I know you're all forgetting about the most important objective of the school, which is our song festival. And you have about 1.5 days left to be prepared. So are you prepared? Okay, this sounds good. All right, very important. Stay on the ball here on our song festival. And then maybe if we have to have a little system to figure out how many contributions we're going to get. So if people already know their grouping, maybe you can send Anna an email to say, okay, this is, we have this country represented. You don't have to tell us what you're going to do, but just let me know which countries are on the list. Yeah.
No, you have to invent. You have to choose your own song. You have to choose your own song. So gather with the people from your country, and decide a song and perform it. No karaoke. Yeah. Well, you can write it on the palm of your hand. Guitar, no, not allowed. No. Yeah. No, it's because guitar is not allowed, unfortunately. They will throw us out of the rest. But you can dance if you don't have to sing exactly. You can dance, you can make other people to sing for you. So you can do all sorts of things. Just a little performance for your country to win a prize. No. Sorry about that. But the wine will help, so no worries. Just some technical things for the students. Please make sure, or for whomever comes tomorrow, make sure you have a badge or you have an identification card, because tomorrow the campus is closed. It's opening practically for us. So breakfast will be served here anyways, but please go and work on your project. We will have a meeting, but not in the auditorium. So make sure you have your badge with you. And for the students, so work already on your projects and organize it for the presentation in slides, with slides. And also for the students that have received scholarship, please start to make a report. No certificate, otherwise you know that. Huh? Yeah, please do. No, I can send you a template if you get lost, no problem. Yeah, great. So do you have any questions? No, okay. So have a happy time. Well, half an hour max for each group. On Friday.